Okay, so let's talk about the fungal kingdom. It's a world of incredible contradictions, isn't it? I mean, these organisms are absolute masters of a double life. On one hand, they're vital to, our ecosystems, our food, our industries, but on the other, they're one of the biggest and frankly most underestimated threats to human health. So in this explainer, we're going to dive right into that duality. We'll explore just how fungi can be both a friend and a fatal foe.
And this just shows that contrast perfectly, right? On one side, you've got the work of Penicillium camemberti, absolutely essential for ripening delicious cheeses like brie and camembert. I mean, that's literally fungus making our food better. But then look at the other side. That's a scanning electron micrograph of Candida albicans, a major human pathogen, forming this tough invasive biofilm. That jump from the cheese plate to a clinical setting, from gastronomy to pathology, that's exactly why mycology is such a fascinating and critical field for you all to be studying.
So to really get a handle on just how serious fungal infections or mycoses are, let's just start with a number. Over 4 million. Just let that sink in for a second. That's the conservative estimate for how many people die from fungal infections around the world, every single year. It's a staggering figure and one that, honestly, just flies completely under the public health radar.
And here's where we get some real perspective. We all hear about the global fight against diseases like malaria, right? And rightly so. It was responsible for about 608,000 deaths in a recent year, a huge number. But look at the number for fungal diseases next to it. It just dwarfs it. We are, without a doubt, dealing with a silent pandemic on a totally different scale. And this right here is why the work you're all doing as microbiologists is so unbelievably important. So that leads to the big question, doesn't it? How on earth are these organisms so good at causing disease? Well, a huge part of the answer is that they are masters of disguise. They're biological shapeshifters, changing their form to get inside the human host and cause havoc. One of their key strategies is something called dimorphism. It literally just means two forms. And for these pathogenic fungi, it's their incredible ability to switch between being a mold out in the cooler environment and then turning into a yeast once it gets inside a nice warm host. This switch, it's a fundamental weapon in their arsenal. It's a key virulence factor. And this diagram just lays it out perfectly. At room temperature, say 25 degrees Celsius, an organism like Blastomyces dermatitis is just hanging out as a mold. But then you inhale it. It gets into your lungs, where the temperature is a cozy 37 degrees Celsius, body temperature. And bam, it converts into its pathogenic yeast form. This change is absolutely crucial for it to dodge our immune system and really set up shop. But, it's not always about temperature. That's not the only trigger. Some of the most infamous fungal pathogens out there, I'm looking at you, Candida albicans, use a slightly different but related strategy. It's called phenotypic switching, and this isn't just a simple mold to yeast thing. This is about changing your shape to do a specific job once you're inside the host. Here, you can see exactly what I'm talking about with Candida albicans. It can float around as a single-celled yeast, which is perfect for spreading through the bloodstream. But when it finds a spot it likes and decides it wants to invade tissue or build a really stubborn biofilm, it switches. It starts growing these long, branching filaments we call hypha.
So let's just quickly break that down. It all starts with the parent yeast cell. It gets some kind of signal from the host environment, and that triggers it to form something called a germ tube. Then that tube just keeps growing, elongating into one of those invasive hyphae. And it's these hyphae that are the real weapons. They're what let the fungus physically punch through host cells and tissues to anchor the infection.
Right, so we've seen how they change shape to get in. Now let's zoom in even closer. Let's deconstruct the fungal cell itself and take a look at the microscopic fortress that makes these things so incredibly resilient. So what is it, really? What makes fungi so ridiculously tough? How can they survive in such harsh places and, more importantly, stand up to the full force of our immune system? What's their secret? Well, the answer, and I'm sure you've already guessed this, is all down to their unique cellular architecture. Specifically, we're talking about their cell wall and their cell membrane, two structures that are fundamentally different from ours. Okay, so let's look inside that fortress. The wall itself is this really complex matrix of polysaccharides. You've got your beta-1,3 and beta-1,6 glucans, which are cross-linked with chitin. Yep, the same stuff in insect exoskeletons. All of that is interwoven with things called manoproteins. And then you look at the cell membrane just underneath and you find this molecule,
ergosterol. It pretty much does the same job as cholesterol in our cells. And every single one of these components is a massive point of difference from our own human cells.
Okay, so here's the crucial part. All this detailed molecular knowledge, we can use it against them. It's time to talk about how we can breach those walls and turn the fungus's greatest strength into its biggest vulnerability. You see, there's a fundamental rule in antimicrobial therapy. Find what's different. And with fungi, it turns out that their greatest strength, that super tough, unique cell structure, is also their greatest weakness. And why is that? It's because that unique structure is the perfect target for our drugs. Think about it. We can design drugs that go specifically after things like chitin synthesis or glucan synthesis or that ergosterol in their membrane, and we don't have to worry about collateral damage to our own cells. Why? Because we simply don't have those things.
And this table just sums it all up nice and simple. Human cells? Do we have a cell wall? Nope. Fungal cells? You bet. That thick wall of chitin and glucans. What about the sterile inner cell membranes? For us, it's cholesterol. For them, it's ergosterol. These are the differences right here that our best antifungal drugs, things like the azoles or the echinocandins, are built to exploit. It's a really beautiful example of targeted molecular medicine in action.
But of course, it's never that simple, is it? Just like with bacteria, we're locked in a constant evolutionary arms race. The fungi are fighting back. They're evolving, they're developing resistance to our best drugs, and completely new, even more dangerous species are starting to pop up. And that brings us to our final and maybe most important question. With these new superbugs, like Candida auris, starting to cause serious outbreaks all over the world, bugs that are resistant to multiple drugs, the need for new therapies is more urgent than ever. So as the next generation of microbiologists, the challenge really falls to you. Where is the next big breakthrough going to come from? The answer to that question is out there, and it's waiting for one of you to find it.